original form because I didn't find that job when I was trying to fill the form. It
Mm-hmm.
was about reagent chemistry manager at Roche.
Oh,
And I started the conversation. I was the first person they started conversation with, but within 2 hours, I came to know that in a meeting they decided to not to list the job until 2020. So that was put off.
oh. Okay, not to give out the job since 2020. Okay, right. All right. Okay. And have you applied to other places?
No,
Like,
I have not applied to other places because my search is only very much centered in Tucson, so I have not applied anywhere else.
no. I mean, like, have you applied like multiple positions for like medical chemist or like any similar position? Like, have you applied to multiple positions or only a few?
No, they had only one position listing, so I applied only to that. I was
Oh,
intending to apply for to this ICAGN position. I think they
mhm.
stopped taking applications already, so that is also not the
Yeah.
option now.
All right. Okay. I understand. All right. So we talked about what did we talk about? Okay. We talked about your day-to-day activities, what you do. We talked about okay, right. So what you do, it's essentially we've talked that it is like target-oriented analysis. Okay, and now we are going to talk about okay. So have you defined the short and long-term scientific strategies as well as like and research goals before like a research project begins?
For this particular project, or in general, you're talking?
In this particular position, like do you define long and short-term project goals or like
Yes,
research
we
goals?
always so we when we say the short-term goals are like SAR development and optimizing drug-like characters, or
Right.
making it more. Sometimes the molecule is big, so you try to make it smaller. So in that way, it's still a generation of SAR and optimization of drug properties.
Right.
So that still goes on. That is always a short-term goal.
Right.
Uh,
But do you define this goal? And do you help like tell the whole team, research team, that this is the goal that we're going to like? This is our research goal. This is what we're attempting to do.
yes, I mean there is a biological team and we are a chemistry team. We always discuss this. Like we are in a DoD project right now, so we always meet once a month, and then we discuss what standing we have. So the biology team gives us the feedback of the previously made compounds and see and gives us which compound is good, which compounds are not. So we come to know based on their assays they do, and then we discuss then chemistry team strategizes how to design further compounds based on the previous results. So do you need
Oh,
improvement? Yes. Do you need improvement in this, or we should keep going in this direction because these compounds are already showing some good activity, or you want to take a whole new path altogether? Because at one instance we had two different compounds in this project. So one was moving forward very smoothly, and the other one was not. We studied them like at least like 30, 40 molecules, and they never went beyond like 10% or 15% of activity, which is not really great. So we
okay.
changed the course of those molecules to a newer heterocycle, and then we started seeing improvement. We identified certain features should be present in the molecule, and then we started keeping those features together and introducing newer features. And so far we have been successful to get good activity. And there are other side activities which we don't want, like scavenging of other radicals or something which we don't want. So that has
Mhm.
been successfully achieved so far.
Oh, that's nice.
Yes,
Okay, so
so we are
yeah,
going
yeah.
in this direction right now.
Okay, so that's okay. So we've talked about like what you do, right? Like
Hmm.
stuff you do, like developing long short-term goals, designing experiments, and the whole like SAR optimization cycle, and developing the structural activity relationships, and
Yes,
like synthesizing compounds, right? So we discussed all of that. Now we're
yes.
going to discuss like the individual research projects that you have done. All right.
Okay,
So
yes.
you over here you wrote that you closely work on the design and synthesis of novel small molecules for mitigating idiopathic pulmonary fibrosis.
That's correct.
So
Yes.
could you start by explaining, okay, what I'm most interested to know is like what were the results of this, and like what was the impact of this, because I'm actually interested in this. I'm actually really interested in this.
Okay. Let me like I was actually I was explaining the same project now, but I can
Right,
get
right.
into more detail now.
Uh-huh.
So this project started some 3.5 years ago,
All right. Okay.
and I didn't work the full 3.5 years, but I worked like 2.5 years on this project.
Okay. All right.
So they were, like I said, there were two compounds identified. One
Mm-hmm.
was a indolin based and one was a purine based. So
Mm-hmm.
indolin based was pursued by a girl, and they gave me the purine based. So I worked on this purine base for like say 6-7 months. I made
Okay.
different compounds, different substitutions. I introduced different functional groups,
Mm-hmm.
different ring systems, but
Mm-hmm.
the activity wouldn't improve beyond like 10 or 15%.
Oh, that
Apart
was the
from
hurdle.
that, yes, that was
That
the
was,
hurdle.
bro. Okay.
Second thing was they used to scavenge a lot of free radicals, which we didn't want,
Okay.
or we wanted it to be a minimal. So these molecules scavenged a lot of free radicals. Secondly, they also had solubility issues. And thirdly, because being purines, most of the anti-cancer drugs have purines, so they used to kill the cells, which we don't want that. So we did like about 2 or 3 rounds of about 5 to 8 compounds each time. So about like 25 to 30 compounds were screened, or even more actually, and we got nowhere. So we had to dump that project. We even screened what is called as bioisoster of purines to replace this purine altogether, but that also did not work. In the meanwhile, the girl who was working, she introduced a ring-like structure on the indolin moiety to improve this, thinking that it will improve the biological activity, but instead it came out to be inactive compounds. So we were scratching our heads, and then after together we put our heads together in a meeting and then said, oh no, we probably there is an amino function, an H function, which is a hydrogen bond donor, that is very important for the activity, because in this the ring in the new analog she synthesized in the nitrogen NH was not present.
Okay.
It was in. So I said, no, we have to introduce this NH back again. So my boss said, okay, why don't you start putting the design and we will order the chemicals. So I put this design together. I found a way of introducing a cyclic ring with
Okay.
a amino function freely available. And then we bought the chemicals, we synthesized, and since then the activities bounced back again. And also,
Okay.
unknowingly, we took care of that scavenging activity also. So there is so far no scavenging activity, and it's not been killing cells.
Oh, and it's not also not been killing cells.
Yes.
Right.
So we improved that property as well. We prevented
Oh,
this being as a cell, what you call as a cytotoxic. We improved
okay.
the scavenging activity almost zero, and we have
Mhm.
sent these compounds to metabolic stability, but we are waiting results. But we are still moving in this direction. So in that way, that was the progress we made.
Right. So this is a project that's currently going on, right? Is
Yes,
it?
this is a DoD project, Department of Defense, from pulmonary fibrosis. Yes.
Oh, all right. Okay. And you have made like how shall I put it? Like how okay, so this new disease that you sorry, this new drug that you've been developing, it's for pulmonary fibrosis, right?
Yes.
And it has this like the hurdles that you faced. It was
Yes.
that one, the activity wouldn't improve beyond like 10%, I believe,
Yes.
right?
Yes.
uh, and like and there was scavenging because of like radicals, and the third thing was it was cytotoxic, right?
Yes.
So you overcame all three of these problems till now, right? All
Yes.
three of these
Yes. Huh?
challenges,
Mm-hmm.
right? Okay. And your contribution, like this was done through team meetings, and this was also done because like you introduced a very innovative design, like a new drug design, right, into the
Yes.
whole
Yes.
molecular structure, right?
Yes. Uh-huh.
Right.
Yes.
Okay, and that was your contribution, like introducing that new molecular structure. correct?
That's correct. Yes.
Okay, I understand. All right. Uh, okay. Closely work on the design synthesis. Okay. Uh, what I also wanted to know is that uh, how big is this research team that you like meet with?
We are three chemistry team, and there is other three people in biological team.
All right. Okay. And have you developed any novel synthetic methods? Like, did you investigate, identify, develop, or optimize any new methods for this?
We usually don't do synthetic methodologies because medicinal chemistry efforts are only put to designing novel, newer compounds, which are
Okay.
totally unknown before. Yes.
Right. Okay. And other than this, did you like how shall I put it? During this whole 3 year long project, did any new information surface from other research that caused you to look at the project again, re-examine the project, and like introduce new changes into the project?
Not at this point, yes. We didn't inquire. I mean, we didn't come across any newer information. It would be really great if they did the protein structure on human uh, cell. I mean, yeah, what is it called? TLR4. Sorry, NADP, human
Mhm.
NADP. They did it on bacteria, which is not really helpful. But there is no development that changed the course of our thinking. No, no, not yet.
Okay, all right, okay. And other than this, let's see here. Okay, and the impact of this research is that this is essentially a drug to, how should I put it, to mitigate the symptoms of pulmonary fibrosis or to like completely reverse it. Like pulmonary fibrosis, it's a type of cancer? No, it's not.
Pulmonary fibrosis is an inflammatory lung disease.
Right, okay.
It is more of due to imbalance between the reduction and oxidation potentials of a cell.
All right.
And due to this, what happens is there is a lot of inflammation in this, and the cells, the tissues in the lung start hardening. And what happens is because of this hardening, the patient is unable to breathe properly. So there is no exchange between the carbon dioxide and oxygen in the lungs. So as this progresses, the patient feels very difficult to breathe, and they are put on ventilators. And usually the patient dies because there are no uh, cure for this yet.
Okay. And this is essentially a cure for this, right?
We are hoping, yes, yes,
Yeah,
it should
okay.
be a
That
cure. Yes,
that's the goal of this. Okay, that's the
yes,
long
yes,
term goal of this, right?
yes.
I
Uh-huh.
understand. Okay. Now, okay, so we've discussed this first project. Now let's move on to the second one. Actively
Okay.
participate in the synthetic chemistry efforts that result in identifying small molecules for the treatment of amyotrophic lateral sclerosis, right?
Yes. Mm-hmm.
Could you like, well, okay, amyotrophic lateral sclerosis. So you're developing a drug to like treat this, right? Essentially.
Yes, yes. To it's
Okay.
ALS. Yes. Uh-huh.
All right. And what was your contribution to this, all of this? And like, what was the like the hurdles that you encountered, and how did you help overcome these hurdles?
There has not been such a big hurdles in this. Uh, we are just in the initial phase of this drug screening, but the thing is, like the
Mhm.
drug in this project, the plus point was that the protein structure which we are targeting,
Right.
the crystal structure is known, and it is available in silico. So my
Um,
boss had done some screening and identified some chemical hits, like usually I talked about previously. So we purchased those, and then accordingly we started introducing newer molecular features to improve the um, what do you say is activity. For now, we are screening these compounds in two. We are directly binding this to the proteins in cell in lab. Not I'm not talking about the computer. In the
right.
lab, isolated proteins in lab. And secondly, there is a fruit fly larva model, where the
Mhm.
fruit fly larvae are genetically modified to simulate ALS conditions, how they happen in
Right.
human beings. So we treat these fruit flies. Once we synthesize these compounds, identify them, we treat these fruit fly larvae to see their turning time. If there is improvement in the larval turning time, that means the drug is very active and showing good activity. So we are able to identify so far 3 or 4 hits, which we are currently trying to optimize for better activity. We have identified newer structural features from our what is called as our deconstructive analysis of the molecules. We identified that we can introduce smaller structures because since this drug have to be introduced into the brain because ALS is majorly a brain disease, a nervous system disease. So we want these compounds to get entry into the brain. So the smaller the compound, the better. If it is become larger, it will be finding difficult to enter. So we identify smaller structural features that can be introduced into the into the molecules. So this part of the strategy is still under study. So we just have found out and we are going to do it now.
Oh, so like what was your contribution towards all of this, towards like finding out these new strategies or developing these new strategies? Were you involved in this, all these activities?
The major proposal what I did is here is we proposed that these molecules as a whole, we tried to break these molecules into 3 parts.
Mm-hmm.
The central core ring and then 2 substitutions on each side. So we truncated each of the substitutions one by one to see which part of the molecule is more important for the biological activity. So in that process, we were able to identify that you don't need one of the structural substitutions is bigger. So that is actually not it's not at we are not at sure because we didn't do the activity on our compounds yet. So but in silico activity and some in vitro assays showed that smaller molecular substitutions on the core ring would be also helpful without damaging the biological activity. So we are trying to introduce that now.
Right, and you've essentially found like how shall I put it, redundant elements to that whole molecular structure, right? Like elements that are not required, like
yes,
right. So
yes.
what you're doing now is that you're removing those elements, and what this will do is that it will make the drug more effective, since like it can like a smaller drug is more effective in entering like the brain cells, right?
That's
in reaching the brain cells.
Yes.
Right, okay. Okay, I understand. Okay, and after this, okay, is there any other thing that you've done over here? Any other like major contribution that you've done?
We have pinpointed a structural feature that might be leading to toxicity. That has been also identified, but it's also under study. We are still developing SAR around that. Essentially, when you treat this drug, we also treat normal larvae, right? So it showed some damaging effect in normal larvae, which should not be the case. If the molecule is to be good, and it is only to be targeted to this genetic modifications in the larva, they should target only that modification, not the normal larva cells. So we had identified a molecule which actually had a feature that could show that it could be toxic. So we are trying to study that as well.
Oh, interesting. All right. And like when developing this whole this whole fruit fly model, like test the effectiveness of the